everyone. Today we're going to see tyrosine kinase receptors, trying to give their activity, some roles, and composition. Okay, let's start with composition. Got here the ligand binding, an extremity and terminal, an H2. Here it is. An extracellular zone, transmembrane zone, juxtamembrane zone, and tyrosine kinase zone, which got two sites. The site of fixation of an ATP molecule and a substrate binding site, and a C-terminal extremity, COOH carboxyl terminal trail. That was the composition. Okay, let's go to steps. We've got here the cytokine binding, here it is, with subunits receptor A and its activation. This one is gonna be activated. Can say that the ligand can bind to the complex of two subunits in case of insulin. The ligand can be hormones, growth factors, or antigen. Number 2, okay, excuse me. We've got the formation of cytokine kinase complex, or we can say association and formation of heterodimer, allowing transformation and recruitments to the membrane. Number 3, we've got associated JAK tyrosine kinase activation, the activation of JAK. Number 4, transphosphorylation of JAK, which phosphorylates the receptor, creating anchor sites for the SH2 domain cytokine protein, which is STAT. STAT is gonna get fixed right over here. Okay. Number 5, JAK phosphorylates STAT. After its recruitment, here is a phosphate molecule fixed on STAT. After we've got STAT detach from receptor and associate to form dimer capable of crossing the nucleus membrane. Here it is, crosses the nucleus membrane. We call this translocation. Translocation. It means changement of location. Okay. Of the dimeric complex, here is the dimeric complex, and its attachment to a DNA traject sequence, not any sequence, a traject sequence, in order to activate its transcription. That's it. Now let's go to see roles. Okay, well, generally, the tyrosine kinase receptor, after having a substrate and an ATP molecule, is gonna add a phosphate molecule to substrate. And we've got here ADP, adenosine diphosphate. Here is 3 phosphates, diphosphate. a protein tyrosine phosphatase is gonna eliminate phosphate from substrate, and we got the initial case. And the tyrosine kinase receptor have to activate number 1, STAT, which is signal transducer and activation of transcription. Number 2, generally it's gonna activate adapter or effector proteins such as PLC, phospholipase C, MAPK, mitogen activated protein kinases, and IRS, insulin receptor substrate. It got many roles such as apoptose, cell differentiation, multiplication, and proliferation. Or it can act on cis and trans, and they are factors of transcription. That was our course today. Thank you for watching, and goodbye.